Hi guys, welcome back. This is Dr. Bueller, and we're going to start our antibiotics coverage by talking about cell wall synthesis inhibitors. Now hopefully you remember a little bit about the cell wall of bacteria. I know if you don't, Dr. Stamper is going to come get you. So we had the inner membrane, we had a cell wall here made out of peptidoglycans, and then we had in gram-negative bacteria an outer membrane. There's lipopolysaccharides and markers and different kinds of lipids in the membranes in different bacteria. And we had efflux pumps and influx pumps and porins and all types of things that are involved in moving nutrients and drugs in and out of the bacterial cell. So bacterial cell walls are an amazing target for antibiotic drugs. And that's because they have wonderful selective toxicity. Again, selective toxicity is simply trying to kill something else without killing ourselves. And the more distant the drug target is from our own enzymes and proteins, etc., the more selective toxicity you're going to have. The best thing about this drug class is we don't even have a cell wall. So if we make a drug targeting cell wall synthesis, it's not going to interfere with any basic metabolic function or structural function in our own cells. So this is a huge class of antibiotics, and you're going to be learning about a lot of different subclasses. So the main ones that you're probably most used to seeing are in Betalactam classes. And the Betalactam classes refer to a set of structurally similar groups that all have this structure here called a beta-lactam ring. And when you talk to Dr. Stamper, he's going to explain all about the beta-lactam ring, and it is beautiful. That is a wonderful ring. But of course, I can't give it what it deserves. You're going to have to wait for Dr. Stamper's MedChem on that. The different kinds of antibiotics that are in the beta-lactam class include penicillins, cephalosporins, carbapenems, and monobactams. We also have some other classes of antibiotics that also prevent cell wall formation, but they do not have a beta-lactam ring, and they don't have the same enzyme drug target. They're going to act at different levels in different places in cell wall synthesis. So these non-beta-lactam classes include a drug called phosphomycin, one called bacitracin, and a group of glycopeptides. So to figure out how these antibiotics block cell wall formation, we have to start by understanding how the cell wall is actually made. The bacterial cell wall is made of peptidoglycans, and so peptido for amino acids or peptides, and glycan for sugar. We're going to see that we have two basic building blocks, NAG, or N-acetylglucosamine, and NAM, or N-acetylmuramic acid. Those are going to be transported outside of the cell itself to the site of the cell wall. NAG and NAM are linked in a carbohydrate backbone, and then those carbohydrate backbones are going to be cross-linked through those peptide chains. That's what's going to provide the structure of the cell wall. So let's look a little bit about how that process actually occurs. First, we start with NAG, and NAG is just the carbohydrate. We're going to take half of that NAG and turn it into NAM. And we're only going to use half because we're going to use the other half of the NAG in its original state. So we have an enzyme called enol pyruvate transferase, and it's going to synthesize NAM by adding some amino acids here. It's important that the last amino acids are two alanines, and Dr. Stamper will talk about exactly how the reactions with those alanines are going to occur. You have the NAG and the NAM synthesized, and you're going to transport them outside of the cell. So that synthesis is occurring in the cytosol. So you're transferring them outside of the cell membrane, inner cell membrane. That's occurring by binding the NAM and the NAG to some lipid carriers that are going to pull it through. I'm not going to go into a lot of detail on how those lipid carriers work, but we do have drugs that can block this step. Once that NAG and NAM is outside of the cell membrane, where the cell wall is going to occur, we're going to see that they're going to be linked together, which if you were thinking about a raft made of logs, you can see here's a backbone here, Here's a backbone here. Here's a backbone here. And so you're going to be seeing these long backbones here. And those are linked together by transglycolases. And here's transglycolase. And you remember what it's linking together because these are the glycol or sugar backbones. Once those are set together, they'll have this linear structure, but there's nothing holding them in place. So you still don't really have a structural cell wall yet. What you're going to do is you're going to link or cross-link those sugar backbones by creating peptide cross-links between them. So the NAMs were the ones that had these amino acid groups. And we've got an enzyme called transpeptidase that's going to come in and it's going to allow the peptide chain from one NAM to attack and covalently bind to the peptide chain of another NAM. Now, Dr. Stamper's going to go through the actual reaction that's occurring there. But what you really want to come away with right now is that it is the transpeptidase enzyme that it's going to create those cross links. There's a lot of difference in terminology. Some people say transpeptidase, some people say penicillin binding protein, or PBP. And that's because the way that transpeptidase was originally identified was by putting penicillin into a bacterial cell culture and then pulling out everything that bound or stuck to penicillin. There were multiple enzymes that stick to penicillin. One of them is transpeptidase that catalyzes this cross-linking. There are some other enzymes that also stick to penicillin. So there's really a whole group of penicillin binding proteins, but since transpeptidase is arguably the most important one in terms of penicillin actions, a lot of times people just use those terms interchangeably. Although you will notice that I couldn't get away from the pharmacology and I had to kind of mention that there are some other enzymes. So let's start now with the major class of cell wall synthesis inhibitors, and those are the beta-lactam antibiotics. And again, there were 4 main classes. They all share that beta-lactam ring, and the molecular and cellular mechanisms of action are that they irreversibly suicide inhibit transpeptidase, or PBPs. The cellular effect of that is that it blocks cell wall synthesis, and what's going to happen after cell wall synthesis is blocked is the bacteria is going to lyse. It's going to explode. The reason for that is just like bone remodeling, where you're always dissolving bone and then remaking bone, dissolving bone and remaking bone constantly. The bacteria are also remodeling the cell wall the same way. So as the bacteria break down cross-links and then try to repair them, they're not able to repair them because they're not able to make these cross-links. Then without that structure in place, the high osmotic pressure inside the bacteria is going to cause it to explode. We will be talking about a lot of beta-lactam antibiotics, and it looks really daunting at first, but trust me, every student before you, every year before you's done this, so you got to just get a memorization system, make a chart, make a graph, make mnemonics, and hopefully after we go through it a little more, they'll start to make sense. One of the things you'll see is that these were created kind of sequentially within these subclasses. So the penicillins themselves were the first ones identified, and that was the whole Fleming and the mold and the bread, or was it bread or was it a petri dish? I can never remember. But those were the first ones identified as they were natural agents. Then med chemists took a look at those and said, hey, we can make them better. And they continued to change those drugs and modify them in ways that we wanted to see clinically. We see the same thing with cephalosporins in that we see these different generations and we change some of the activity of them as we go through the generations. One of the things you'll see in the antibiotics lectures is a visual to try to talk about whether things have a lot of activity at gram-positive or gram-negative. So in the case of the first generations, we have a lot of gram-positive activity, but very little gram-negative. As they synthesized new ones, we went down through the generations, and you got less gram-positive, but more gram-negative activity, until you reached the third generation, in which you had a lot of gram-negative and little gram-positive. Then clinicians found a new need in cephalosporins, and those were ones that were going to be more targeted toward MRSA and Pseudomonas. So med chemists came in and they created some more generations that were pretty evenly active against gram-negative and gram-positive. So they're pretty broad spectrum, but they were really focused specifically on these two bacteria. Some basic things to remember for all beta-lactam agents is that because they share the same basic structure, they're going to share some really important properties. One of those is cross-allergy. So a beta-lactam ring itself isn't antigenic. That means it doesn't create antibodies. But the beta-lactam is what we call a hapten, which if it's complexed with a larger protein, like albumin in your blood, that whole thing, so let's get albumin here, that whole thing then can be identified by your immune system and you can create antibodies against it. Once you've created an antibody against this beta-lactam ring, any other drug that comes in that has that same beta-lactam ring easily accessible is also going to be identified by the antibody. So if you take a penicillin and you create an antibody here against that beta-lactam ring and become allergic to it, and then you want to switch over and take a different penicillin, you're still going to recognize that ring. And that's what we call cross-allergy. Now, maybe then if you take a carbapenem or a cephalosporin, your antibody might recognize that beta-lactam in those drugs as well. So whenever we have an allergy to a beta-lactam ring, you have to at least be thinking about the fact that you could have cross-allergies with any other drugs that are in the beta-lactam class. The allergy with beta-lactams is really specific. A lot of people get a little bit of a rash when they take beta-lactams that isn't a true allergy, and it's not something that you're really concerned about. When you see a true allergy with beta-lactams, there's a really specific phenomenon called socks and gloves, and that's a rash that occurs on your hands, it's like from the wrist down to your fingers, and on your feet from your ankles down. And it's red and it's swollen and it itches, but it's really focused on those regions. Don't know why. But that rash signifies that you have a true immune response to that beta-lactam. If that immune response is too high, then upon reintroduction of beta-lactams, you could have anaphylactic response. And that's why with beta-lactams, a counseling point is always to ask about, do you have any allergies to other penicillins? I say here that each drug class has a different risk, and that's because, as Dr. Stamper is going to show you, that beta-lactam ring can be sticking out more easily to be found by an antibody, or it can be hidden a bit more and less likely to be recognized by the immune system. So these beta-lactam antibiotics seem amazing. They block cell wall synthesis. We don't have cell walls, so we have selective toxicity, so that's great. These should work for everything, and they don't. So we started out, we had our basic penicillin, and everything looked good. The bacteria, they make those PBPs, including transpeptidase, and we figured we can target that transpeptidase, and we're going to create a drug that's going to be a beta-lactam antibiotic. So us wonderful basic sciences folks here create this drug. Then, through mutations, bacteria can gain resistance to these beta-lactam antibiotics. And one way to gain resistance to these is to break up that beta-lactam so it no longer works and it can no longer bind to the transpeptidase. So many bacteria have now made an enzyme called Betalactamase. So it's the beta-lactam, ACE, it breaks up the beta-lactam. So now we've got the bacteria, they have the PBPs, we've created beta-lactam antibiotics to block those PBPs or transpeptidases. Now the bacteria make beta-lactamases to break down our beta-lactams. So what are we going to do? Well, a med chemist could come in here and say, we've got this beta-lactam ring, it's being broken down, let's change this drug structurally so that the beta-lactamase can't reach in here. So they've made drug classes that are more beta-lactamase resistant. A pharmacologist may come in here and say, well, you've got the beta-lactamase, so let's use that as a drug target, and let's create a drug that inhibits the beta-lactamase. So we use both strategies. We've created beta-lactam drugs that are less susceptible to beta-lactamases, but we've also created drugs that block the beta-lactamase itself. So now you've got bacteria with transpeptidases. We're blocking those transpeptidases with beta-lactams. Bacteria then have created a beta-lactamase to break down the beta-lactam, and we then create a beta-lactamase inhibitor to block the beta-lactamase. We have several beta-lactamase inhibitors, and what you're going to see is you're going to see these in combination with the beta-lactam antibiotics. So the first one is clavulanic acid. We also call it clavulanate. Then you'll see sulbactam, tazobactam, avibactam, and viborbactam. Each bacterial strain that created beta-lactamases created somewhat different beta-lactamases. So there are multiple different types of beta-lactamase. So when you see a beta-lactamase inhibitor like clavulanic acid, it may inhibit some forms of beta-lactamase but not others. So you're going to see that different beta-lactamase inhibitors are going to be used in different infections. Mechanism of action, really important. These are not antibiotics. They do absolutely nothing to block bacterial growth. They don't kill bacteria. They do nothing in and of themselves to bacteria. All they're doing is halting the destruction of your antibiotic. So the antibiotic is the antibacterial, and the beta-lactamase inhibitor is just helping the antibiotic not be broken down. By the end of this section, you're going to see several different drug combinations, and you're going to be able to identify the agents in those combinations are. You're going to see augmentin, which is amoxicillin and clavulanic acid. You're going to see unicin, which is ampicillin and sulbactam. You're going to see zosyn, which is piperacillin and tazobactam. And you're also going to see timentin, which is ticarcillin and clavulanic acid. The way I remember which beta-lactamase inhibitors in zosyn is it's that tazobactam.